Today I will teach to you in this video is the next point in this chapter is the estimations of hardness by EDTA methods. In previous video we have studied the types of water. Waters are mainly classified into two types, soft water and hard water. What is by soft water? The water which do not contain any dissolved impurity of a calcium and magnesium salt. Such salts are called as the soft water. And hard water means the water which contain the salts of calcium and magnesium in the dissolved form. Such waters are called hard waters. I understand. How will you calculate this hardness present in the waters? The hardness present in the water is estimated or calculated by using the EDTA methods. Therefore, try to understand the estimations of hardness by EDTA methods. EDTA is a chemical compounds. Na2 EDTA means disodium ethylene diamine tetraacetic acid. What is the long form? Disodium ethylene diamine tetraacetic acid. In these chemical compounds, they contain the 2 sodium atom and 3 other molecules. These 3 molecules always complete in the 10th class. Ethylene, you know the formula of ethylene which is completed in the 10th class is the CH2 double bond CH2. Amine group, functional group of amine is the bond NH2. It is also completed in the 10th class. Acetic acid also completed in the 10th class. The formula of acetic acid is the CH3COOH. And you know the sodium means Na. Disodium means they contain the 2 sodium atom. Tetra means 4 acetic acid molecules. How will you draw the structure of this Na2 EDTA? First, the fundamental molecule is the ethylene. First, draw the structure of ethylene. Take the molecule of ethylene, CH2 double bond CH2 the ethylene. When the 2 molecules of amino group NH2 group join to the each carbon atom, this is the NH2 joined to the one carbon atom and the another NH2 joined to the another carbon atom, remove the double bond. This is the structure of ethylene diamine. It contains the 2 amino group. Amino group is 2. Ethylene diamine, ED. Ta means tetraacetic acid. These 2 hydrogen atom of amino group replaced by acetic acid CH2 COOH and remaining another hydrogen atom of amino group replaced by another acetic acid group. Here also these 2 hydrogen atom replaced by 2 acetic acid group. And it is the structure of ethylene diamine tetraacetic acid, but they contain the disodium. Therefore, this 2 hydrogen atom of acetic acid group is replaced by 2 sodium atom. Then we get a structure of Na2 EDTA. This structure of Na2 EDTA, disodium ethylene diamine tetraacetic acid. I understand. You know that in the amino group nitrogen atoms have the lone pair of electrons. Is it not? This is the structure. This EDTA, disodium ethylene diamine tetraacetic acid, is a highly sensitive because they react with a heavy metal which is present in low concentration in the waters. They react with high concentrated metal also, as well as they also react with the low concentrated metal present in the hard water. Therefore, in the titration, EDTA aqueous solution is used. This is the structure of EDTA. This is the theory part. I understand. How will you, when this ethylene diamine tetraacetic acid is reacted with hard water sample, in this titration, generally 2 points is very important because EDTA is react with hard water sample at a definite pH. Therefore, use the buffer solution here. What is the buffer solution? What is meant by buffer solution? Here buffer solution whose pH is 10. Add the 4 to 5 ml of buffer solution. What is by buffer solution? The solution which have ability to change in pH when you add small quantity of acid or base. It means that if you take the buffer solution, any buffer solution whose pH is known, add small quantity of HCl, acid, and check the pH. pH do not change. Take same pH solution in another test tube and add small quantity of base like NaOH. Again change the pH. pHs do not change. Such type of solutions are called as the buffer solution. The solution whose pH remains constant after adding the small quantity of acid or base. Such type of buffer solution taken and add in the hard water samples and use the EBT, Erichrome black tea indicator. Add the 4 to 5 drops of EBT indicator. Then what happens? You know that EDTA is highly reactive. They react with a calcium or magnesium present in the hard water and it form a complex compound. Therefore, this titration called as the complexometric titrations. How it is formed the complex? When this EDTA added in the water sample which contain the buffer and EBT, it form a complex compound. What is the structure of complex here? When you add the drop by drop EDTA, then what happens? It form a bond, oxygen atom of carbonyl group form a bond with a metal and the metal form a coordinate covalent bond with a nitrogen atom. Similarly, the another carboxylic group CH2 C double bond O CH2 CONE. Here also the nitrogen atom oxygen atom form a bond with metal and this metal form a coordinate covalent bond. The metal present in dissolved form water form a coordinate covalent bond with two nitrogen atom. This is the complex compound. Colorless complex compound is formed here. Understand? This is the theory parts of these titrations. How will you calculate actually in the laboratory? This is in laboratory, the EDT method is divided into two parts. In the first part, the standardization of EDTA solution. What is the standard? What is by standard of EDTA solutions? Calculate the molarity of EDTA solution by using the known molarity of another solutions. Therefore, first make the standard zinc sulfate solution, means whose molarity is known, and pipette out, take the 10 ml of zinc sulfate solution, pipette out the zinc sulfate solution, aqueous zinc sulfate solution, and take the 10 ml of zinc sulfate solution in the conical flask, add the 4 to 5 ml buffer solution in the zinc sulfate solution, and then add 4 to 5 drops of EBTA solution in this zinc sulfate solution. Then it form which color? Pink color is formed. Which color is formed? Pink color form to the zinc sulfate solution by the adding the EBT indicator. Titrate this pink red color zinc sulfate solution or mixture with a EBTA. Fill up the EBTA solution in the burette up to the mark and add the EDTA solution drop by drop in the zinc sulfate solution until the color of solution changes from pink red to sky blue. Here sky blue color is formed. This is the end point of this titration. Then note the reading, burette reading. Consider this is the VMN or Note the reading and calculate the molarity of the EDTA. EDTA titrated against zinc sulfate solution. Therefore, M1 V1 is equal to M2 V2. Here M1 is the molarity of EDTA. V1 means burette reading, volume of burette. M2 is the molarity of zinc sulfate is known. V2 means 10 ml pipette solution. Therefore, M1 is equal to M2 V2. Value of M2 and V2 is known and V1 is known, and calculate the M1. This is the part first. Then do the part second. Then calculate the total hardness present in the water. Here, take the sample solution. Pipette out the again sample hard water solution. Take the 10 ml pipette with the help of pipette in the conical flask. Again, add the 4 to 5 ml of buffer solution in the water sample, hard water sample solution, and 4 to 5 drop of EBTA solution. Then what happens? Actually, what happens here? What is the reactions? When you take the water sample, consider the water sample containing the calcium ion. If you add the EBT indicator and buffer solution, then this EBTA is react with calcium and it form a calcium salt and 2 hydrogen ion is removed, which having the pink red color. This pink red color solution titrate against EDTA solution. You know that EDTA is highly sensitive chemical compound. They react with the hard metal present in the water and form a complex salt of a EDTA, and color of solution is changes to sky blue. This is the end point. Then in the pink color solution, add the again fill up the burette up to the 0 mark and add the drop by drop EDTA solution in the water sample until the color of solution it changes totally sky blue. Sky blue color is formed to the solution, and note the reading again. Consider this reading is the YML. Then calculate the total hardness present in the sample. Total hardness is equal to Y upon V into M1 into 100 into 1000. What is the Y here? Y means volume of EDTA, that is the burette reading. What is the V here? V is the volume of sample. How much? 10 ml. M1 is the value is calculated in the part first. Substitute the value and calculate the, consider this total hardness is a Z. Is a Z. Z ppm calcium carbonate equivalent. We get a total hardness. By using the same EDTA method, we can easily calculate the temporary and permanent hardness. How will you calculate? This is total hardness, means it contains the temporary and permanent hardness. Then how will you calculate temporary and permanent? Take the same sample solution, boil it. You know that temporary hardness water means the water which contains the calcium or magnesium bicarbonate. This temporary hardness is destroyed with the merely boiling. Therefore, boil the sample and remove the temporary hardness and filter the solution. The filtrate contain only permanent hardness. Take this 10 ml of permanent hard sample solution and again add the buffer solution and EBT indicator and titrate against the EDTA solution and calculate the permanent hardness. Calculate the permanent hardness. Consider this permanent hardness is AML. A. Then How will you calculate the temporary hardness? Temporary hardness is equal to total hardness minus permanent hardness. Means this is the Z minus A is equal to temporary hardness. In this way, by using the EDTA method, we can calculate total hardness, permanent hardness as well as temporary hardness. Which type of question will be asked on this point? Describe the estimations of hardness by EDTA method. Such type of long question will be asked in the examination, or some multiple questions or problem is also asked on this point. That question as well as some important point of this video will be given in the description box. Thank you.